Good morning, students. Today, I'll be discussing with respect to the oncogenetics. So, as we all know very clearly, the subject oncology is very wide and also involves a lot of aspects. In that, one of the main feature which is involved is the genetic factor. So, hence, it is an entangled word, oncogenetics. So oncology, or you can say cancers, is nothing but it's a disorder which is because of the abnormal expressions of the genes. Abnormal expression of the genes, which may result in abnormal cell division. Okay, so hence, whenever we have to study oncology, we also have to study the genetic features of that particular oncology, or the gene which is responsible for that particular cancer, which we call it as an oncogene. Yeah. So, as I was telling, cancer is a name used to describe the virulent forms of neoplasia. Neoplasia, that is uncontrolled proliferations of the cells. So once the cell starts to divide, they will not stop the division, which results in forming a tumor or a neoplasm. Okay, so neoplasm, a new type of cytoplasm exists which causes the neoplasia. Is that clear? So if this neoplasm should be, if at all, if neoplasm to be considered as cancer, it has to become malignant. So malignant means it will develop a feature or a character where it starts to, the cell which has undergone change will start to influence the other cells and make sure that the new originated cells are all getting converted into a malignant cell or a neoplastic cell. That is referred to as malignant, and this malignant will result in creating cancer or originating a cancer. Is that clear? And the process is referred to as metastasis, and all of us are very familiar with the terminology metastasis. So this abnormal accumulation or imbalance between the normal cell proliferation and cell regulation is always under the control of a mechanism called as programmed cell death. So every time a cell loses its capacity or energy, that cell has to be removed from the body. And that mechanism is genetically done, and that is referred as programmed cell death. Okay. Now, if these cells are not properly removed, then the possibility is that these cells starts to abnormally divide and result in creating cancers or neoplasms. So ultimately, as we define, cancers are due to abnormal expression of a particular gene, which results in causing a genetic disorder, and this disorder is due to the due to the reason that there is no repair mechanism working properly in those cells. So there is always a genetic repair mechanism which works to remove the cells from the body, and if those genes gets mutated, the probability is that this cells becomes cancerous in nature. Is that clear? So, and these, even though we call them as genetic disorders, they rarely get inherited as they are, but few of them carries to the next generation, and most of them, they are somatic in nature. Is it clear? So these cancers are divided into 4 classes. First is the sarcoma, carcinoma, and lymphoma. These are the and leukemias, lymphoma and leukemias. Of course, in the lymphoid system itself, there comes the leukemias. So basically, we classify them into three main types and one sub-main type, that is sarcoma, carcinoma, lymphoma, and leukemias. Okay, so these are the four classes. First is the sarcoma, is the one which arises in the tumor, which we normally come across all over the body in many cases. A few of them, all of us, most of them will have one or the other type of these mesenchymal bulges in the body, and they are restricted to skin or mesenchymal tissues, and they are non-malignant. So because of that, they will be left out all through the lifetime. So they may not become dangerous at any point of time, but very rarely they can also get converted into cancers. If converted into cancers, resulting in affecting the bone, muscle, or connective tissue, or in case of nervous system. Carcinomas, whereas in the other hand, carcinomas originate in the epithelial tissues, such as the linings of the intestine, bronchi, or the mammary glands. Whereas the hematopoietic and the lymphoid malignant neoplasms, such as leoplasms, will form the lymphomas and the leukemias, which in turn is due to the abnormal multiplication of the B cells or T cells or the RBCs that results in causing leukemias. Okay, and these leukemias are faster in spreading and they kill the body much faster than the sarcomas or the carcinomas. So the basic properties of a cancer cells. So here we have to note at least 10 different points which will make us understand what happens, what is the characteristic feature of a cancer cell. So for that, we have to have a distinction between the cancerous cells and the normal cells. So normal cells usually do not divide unless they are stimulated to do so by the body's homeostatic mechanism. Okay, because as I said, the repair mechanism is well in place and they have to manage the division well. So because of that, in the normal cells, only when there is a certain level of homeostatic balance, the cell undergoes division, and once they divide, they stop their division. Whereas in contrast to the normal cells, in case of most cancerous cells experiences a breakdown, where breakdown of the regulatory mechanism of the division, which influences the self-destruction. So the repair mechanism doesn't work. In those cases, there is a possibility the cells will undergo formation of tumorogenesis, or you can say cancerogenesis. Okay, and these cells proliferate and results in creating cancers. Now, when cancer cells are grown in tissue, if in vitroli grown in tissue under conditions that promote cell proliferation, they grow and divide at a rate similar to that of their malignant counterparts, forming multi-layers. Okay, so one layer, there is a specific arrangement of cells and there is a specific interaction between the layers of cells. Whereas here they don't, they just form a monolayer and there is no further organization of the cellular mechanism or the tissue gradation, which results in abnormal accumulation of the total cells. Okay, so resulting in cancerogenesis. Then the inhibitory, or you can say the growth rate, the division rate of the cells immediately drops as it is under the normal inhibitory influences from the neighboring environment. Whereas in case of cancer cells, these cells continue to pile up and absorb the nutrition and start dividing more and more and start accumulating themselves. Next, growth inhibiting influences may arise due to the result of depletion of growth factors in the culture medium or from contacting with the surrounding cells. So in malignant cells, they are not responsive to the types of signals that cause their normal counterparts to cease the growth and division. Next, cancer cells ignore the inhibitory growth signals. That is, the cell death mechanism is cancer cells ignore the cell death mechanism. As I said, the signals are not caught, or you can say not observed or received properly by the cancer cells. Because of that, the cell death will become abnormal, so the proliferation continues. Whereas in the normal cells, the signals are clearly received and the cell division stops because of the normal interaction of the cells. Okay, so cancer cells are seemingly immortal because they continue to divide indefinitely. Okay, their difference is growth potential is often attributed to the presence of telomerase. So all we say is telomerase is nothing but it's an enzyme which is very important to maintain the ends of the chromosomes, allowing the cells, thus making sure that the chromosomes are well under proper regulation or protection. But in cancer cells, what happens, as there is repeated division, there is no time for the telomerase to act. So hence, every time the chromosomes divide, they lose a template, or you can say a fragment of DNA, which will be used by the RNA template for DNA synthesis. So the absence of telomerase from most type of normal cells is thought to be one of the body's major defenses that protects against the tumor growth. Okay, so types of normal cells is brought to be one of the body's major defenses that protects against the tumor growth. The most striking alterations in the nucleus formation is transformation occur within the chromosomes. Unlike normal cells, that replicative DNA is very less in correcting, or you can say low error rate. Cancer cells are genetically unstable and often have highly aberrant chromosome complements. So in the next class, I'll be detailing about the chromosomal basis of cancer. So you people will be knowing very clearly how exactly the chromosomes are involved in causing the cancers. Cancer cells typically fail to elide the apoptotic response, as I was detailing again and again, the same thing. Okay. Next, with respect to the energy usage, the cancer cells often depends on glycolysis, which is considered as an aerobic metabolic pathway. This property may reflect the high metabolic requirements of cancer cells and inadequate body supply within the tumor. However, when the oxygen is plentiful, many tumor cells continue to generate much of their ATP by glycolysis, called aerobic glycolysis. So under conditions of hypoxia, reduced oxygen condition, cells activate a transcription factor called as HIF that induce the formation of new blood vessels and promotes the migratory properties of cells, which may contribute to the spread of tumor. So this is the main reason for malignancy. That is, the malignant cells is due fastens when there is hypoxia, there are the reduced oxygen rate. Even though glycolysis actually generate less ATPs comparing to the oxidative pathway or the Krebs cycle pathway, in mitochondria, it produces ATP at a more rapid rate when they are in cancerous condition. The increased uptake of glucose by tumor cells compared to normal cells can be used as a means to locate the metastatic nature of that particular tissue or organ. Is it clear? So these are the important features of the cancerous cells, which we have to note them. So I'll be detailing the mechanism of cell cycle here. So how the mechanism of cell cycle influences the rate of division of the cells, and in turn may result in creating cancers. So the interface, that is the cell cycle is divided into 4 phases, that is G1, S, G2, and M phase. We all know this. These 4 phases, the interface which starts with the G2 phase. Okay, so G2 phase, there in any cell cycle there are 2 important components which are involved in cell division, that is cyclins, okay, and cyclin dependent kinases. 2 important components are involved in cell division. One is cyclins and cyclin dependent kinases. So you can see the level, or you can say the concentration of these 2 components interacting from these 2 components, which carries the cell division in a proper rate. Okay, so the interaction between the cyclins and cyclin dependent kinases. So here you can see the cyclin B, that is the cyclin B accumulates through the interface near the end of G2. Okay, cyclin B accumulates near the G2 phase, G1, S, G2, and M phase. So it's the end of the cell cycle which is starting, we are taking, because as I said, cancers are mainly due to the loss of stop of the cell division. That is, either they have to apoptotically be strong or their repair mechanism should be very strong. So in the G2 cycle, there comes the two important repair mechanism, that is the P53 and P21, which mainly works here. So the cyclin B accumulates throughout the interface near the end of G2. Active MPF, mitotic promoting factor, reaches a critical level which causes the cell to progress through G2M checkpoint and into mitosis. Is that clear? So very important. Before going to mitosis, it is necessary that all the total amount of DNA is properly produced, and that amount of DNA should be sufficient enough to form the normal chromosomes. So now to maintain that, the cells has to be altered and checked, and that is done by the cyclin B. Now, as you can see in the cyclin B, okay, cyclin B, as you can say, the level of active mitotic promoting factor. So you can see that increases by the time of mitosis. So the degradation of cyclin B near the end of mitosis causes the active MPF level to drop. So the mitosis completes and the mitotic promoting factor is red. Mitotic promoting factor concentration comes down, so that the cell will not enter into the G1 phase for further synthesis of the components necessary for the mitosis, next cycle of mitosis. So the increase in level of cyclin B during the interface takes place, so that it combines with, as I said, the CDK cyclin dependent kinases is more to produce increasing level of inactive MPF. So more amount of inactive MPF is observed at this region. But by the time of interface, as we enters into the interface after completing the G phase, the end of interface, active factors remove phosphate groups from MPF, producing active MPF, mitotic promoting factor, which brings about the breakdown of the nuclear envelope, chromosome condensation, spindle assembly, as we know with respect to the normal cell division and other events associated with mitosis. So immediately there is again rise in the mitotic promoting factor, and the mitosis takes place. After mitosis, normal mitosis, the mitotic promoting concentration comes down, whereas the cyclin B dependent kinases rises and make sure that the cells are properly arranged with respect to anaphase, telophase, and the cytokinases takes place and eventually enters into back into the interface cell. So this is how the cell cycle plays its role. That is the two important components, that is the mitotic promoting factor and cyclin B, co-regulate the cell division mechanism. Is it clear? So let's this is the aspect or concept which we have to know very clearly with regard to the conversion of normal cell to a cancerous cell, because this is where the changes in this mechanism will make the cell to enter into a abnormal division condition. Okay, so I'll end up here. So thank you.